in your belly.
You're on the shows, the water breaks, okay, that's everything basically that's protecting the baby. That's one of the very first objectives. We've got to know the function of amniotic fluid.
I feel like he's just as pooped as the rest of us. You can just turn it all off. I don't care.
Yeah, that's awesome.
Yes,
Yes, that's better.
all right. You guys know I always tell you, I'm truthful with you, I've never tested amniotic fluid ever in my career. Okay? They do not, a lot of times, run tests on amniotic fluid without it being sent out. The kind of test they do on amniotic fluid generally is done by reference lab. Now, there's some stat tests that they do on amniotic fluid that we'll talk about later. For example, if a woman is rushed to the ER and they're not sure if her water broke or if her bladder is ruptured, they can do a test that we'll talk about called a Fern test. That's what comes to my mind, where they take a glass slide and they just put a drop of the fluid on the slide and they let it dry. And if it looks like a fern, like crystals, a plant, a fern, that's amniotic fluid. Okay. If it's not, then it's probably urine from the bladder. That's one of the few tests that I really think that they want to know stat is a Fern test, and that will be in this PowerPoint as well. Okay, so physiology. The amnion is a membranous sac that surrounds the fetus itself. Within that sac contains the amniotic fluid. There's about 4 or 5 functions of that. A lot of these make common sense, so I don't think you'll have to, really go deep into this. It provides a protective cushion for the fetus. It allows fetal movement to occur. It also stabilizes fetal temperature exposure. It permits proper lung development. We're going to spend a lot of time today talking about lung development and all of these different tests that can be done on the amniotic fluid to determine if the baby's lungs are mature enough to do a preterm delivery, to do an early delivery. There are exchanges of water and chemicals among this fluid and maternal circulation. So here's a picture. Here's the amnion right here. As you can see, all of this would be the amniotic fluid. There's the umbilical cord. Here's the placenta that I was telling you that I've seen many of these, okay? And those little deals, I would call those like veins that are in it. Have any of you ever seen a placenta, ever?
Yeah, it's just got snake side, it's got a red side.
I think in some countries it's a delicacy and they actually eat it.
Yeah.
You can't consider it killed.
What?
Yeah, but
Oh, you have?
Some have 2 quarts and that's 3 quarts.
I didn't know they'd show it to the female.
Yeah, they gotta put
I
it in
think what they do with it, it's not a delicacy. They just
Oh, okay. So you guys got a pretty good idea where the fluid is. This is where that needle is. They do the amniocentesis. It's usually guided with like some sort of an X-ray where they're really sure about where they're at. But the fluid they obtain is this fluid right in here. So they got to be real careful placing that needle. Okay.
Right.
I see up there on the picture, it says chorion. Is that connected to HCG in any way? Human chorionic gonadotropin?
Yes, it is. All right, the volume of the fluid. This may interest you guys a little bit. The volume of amniotic fluid that is within this little membrane, a lot of it actually comes from fetal urine. Okay, the baby is avoiding urine a lot, and they can actually determine how far along someone is during pregnancy by the amount of creatinine and BUN that's in that fluid, because we know there's a lot of BUN and creatinine in urine, right? And so they can determine that. Now there's also lung fluid, and there is fluid that is from the maternal circulation within the amniotic fluid. It says during the first trimester, approximately 35 mils of the amniotic fluid is derived primarily from the maternal circulation. But it says increased amniotic fluid will get up to 800 to 1200 mils in the 3rd trimester, and this is usually the result of fetal urine production. says increased urine is regulated by increased fetal swallowing. So the baby's swallowing the urine, okay? That kind of interested me the first time I really got into reading this. Says lung fluid adds lung surfactants to amniotic fluid, and this is used as a measure of lung maturity. That's probably the most important point we're going to hit about this chapter, is the test that has to be done on the fluid to determine if the lungs are ready for delivery.
So in that test, we're looking at the amount of lung surfactant.
There's about 5 different tests you can do for that. Jason just informed me, I had no idea this, and I don't know where Jason studies a lot, other stuff, but he told me that now that they've got things that they can do at these children's hospitals and places like that, to where they can inject lung surfactants, like artificially.
Wow.
I didn't know that, but he was telling me they can't do that to help the baby be delivered early.
I know they can give you medicine to mature, I didn't know that.
Anyway, you guys can study, I didn't know it, I had no idea, I'm gonna probably have to look some of that up. Polyhydramnios, that is an excessive amount of fluid from failure of the fetus to swallow. Sometimes if the fluid gets greater than 1,200 mils, the definition or the term for this is called polyhydramnios. This can also be due to neural tube disorders, chromosomal abnormalities, infections in the fetus, and even cardiac arrhythmias. Now, if there's a word for too much fluid, there's a word for not enough fluid, okay? And that's gonna be, I think I'm pronouncing it right, oligohydramnios. Says decreased amniotic fluid from increased fetal swallowing. Or you can, of course, have a membrane leakage, which you really don't want to happen. And that's gonna be if you've got less than 800 mils. The composition of the fluid, it is very similar to the maternal plasma or the mom's plasma, except it will contain sloughed off fetal cells. That's what they do the cytogenetics on. If you've ever wondered how, I mean, this is just the basics anymore, but when I worked in the lab, it was considered a big thing, but those fetal cells is what they can do genetic testing on, and they can right away determine if the baby is male or female, for example. That used to be a big thing. Now that's nothing. They can determine that, just on the ultrasound and all of that. But those fetal cells, they can actually test those fetal cells, and that's where they can determine if, unfortunately, that baby's gonna have issues, okay? And by issues, I mean things like Down syndrome. They can tell long before that baby's born if there's something wrong with that baby. And that is a major reason why they do amniocentesis procedures. I mean, you don't really want that done, okay? Because it's kind of, I'm not saying it's dangerous because they do it a lot, but a normal term delivery, there's not a need to do an amniocentesis. But if you have a female that is possibly, let's say that they've had a baby before and it had a genetic abnormality, then they're probably gonna do an amniocentesis. And I think in the next couple of PowerPoints, it's gonna tell us all the different reasons why people have to have amniocentesis done. Sometimes they have to have it done just because the female is over 40, okay? And they wanna make sure everything's going good with the delivery. But there's lots of different reasons that they do amniocentesis. It says the fluid contains substances that are produced by the fetus, such as bilirubin, okay? We're gonna talk about hemolytic disease of the newborn today, and bilirubin plays a major role in that. It also contains lipids, enzymes, electrolytes. The fluid also contains urea and creatinine. It says as fetal urine increases, so does creatinine, urea, and uric acid. Remember that? BUN, creatinine, uric acid. Those are the non-protein nitrogens that we studied in that chapter. Fetal age can be estimated by the amount of creatinine that's in that fluid. Less than 36 weeks gestation, the creatinine level is going to be 1.5 to 2 milligrams per deciliter. After 36 weeks, it'll be greater than 2, because a full-term delivery goes 40 weeks. When I first started teaching, I had to have a student tell me that. I thought it was 36. Because I was doing the math, 9 months times 4 weeks is 36. Actually, a full pregnancy goes 40 weeks, not 36. Just thought I'd let y'all know so you don't get that mistake.
Our youngest was born 36.
It's on 12, sure.
Okay, maternal urine versus amniotic fluid. A lot of times we have to differentiate again, are we dealing with amniotic fluid or are we dealing with urine from the bladder? This test can be done to determine a premature membrane rupture or an accidental puncture of the maternal bladder versus amniotic fluid. Things that you're gonna wanna measure, amniotic fluid will have BUN and creatinine in it, but it'll have less than 3.5 milligrams per deciliter of creatinine and less than 30 milligrams per deciliter of urea. If it is truly maternal urine from the bladder, the creatinine levels are gonna be sky high, okay? So, I mean, that's how you can differentiate it. It says values as high as 10 for creatinine and 300 for urea may be found if the sample is actually maternal urine. Does say the presence of glucose, protein, or both is associated more closely with amniotic fluid. Here's that test I was telling you about called the Fern test. That's where you actually take one drop of the liquid and you just you can take a swab, you can smear it onto a microscope slide, and you just let it dry. And you look for fern-like crystals. It'll look like a fern leaf, okay? And for some reason, and I was talking to Jason about this before class today, they do this test all the time at National Park Hospital. They want to check and see if it's amniotic fluid. I never once did this test at St. Joe or CHI, and I don't understand why, because why would they both deliver babies, and I would think that both of them would do it all the time. But if you go to National Park Hospital, you will see this Fern test more than likely.
If we do a test and we're looking for the crystals and we don't see any, does that tell us that we may have had a messed up amniocentesis?
Well, a lot of times if a female comes into the ER and they think their water broke, okay, for example, they can take a drop of that fluid and find out, is that truly amniotic fluid, or, you see what I mean? Because when a woman goes through pregnancy, you also have to worry about, it pushes on the bladder, okay, so they can kind of urinate on it, so I guess. All right on that?
Yeah, see, the Fern test ain't the only thing they're gonna do, they got litmus paper too that they can test the fluid with, and that gives you an indication if it's amniotic or not.
That's exactly what Jason told me. Jason said a lot of times they can actually test the pH of the fluid also, and tell you a lot of information.
As far as the amniocentesis is concerned, aspirating fluid from out of the mom's womb, or et cetera, that stuff should always have Fern.
It should be, but you gotta remember that amniotic fluid is not far from where the bladder is.
Right.
And so if they were having to do that emergency and didn't have to do that like CT guided or with the ultrasound where they can tell exactly where that needle is, I'm just saying, I don't think it would ever happen, but sometimes there could be an OBGYN saying, I don't know if this is amniotic fluid or urine. I want to find out real quick. And so they can just put a drop of that on a microscope slide and look for those Fern crystals. But like Wendy said and Jason told me, that's not the only test they do to determine whether it's urine or amniotic fluid. And if you guys will remind me, I've got it on a flash drive, but I've got a picture of these fern-like crystals. But if you want to go to YouTube and Google images, you'll see picture after picture after picture. All right, so why do they do amniocentesis? The indications for it: abnormal screening blood test. If they do, most of the time during a prenatal workup, they are going to do an alpha-fetoprotein test. That's called an AFP on the mom. Okay, this is not on the amniotic fluid. This is on the mom's blood. Like when they go to their OBGYN at so many weeks, they'll do a test called an alpha-fetoprotein. If that test is elevated, that can indicate that there's problems with the baby, all right? And then that would give them an indication we need to do an amniocentesis. Now this word human chorionic gonadotropin, now you guys should associate that with pregnancy, like an HCG test, all right? That's the hormone they're testing for pregnancy. But there is a test called a beta HCG, okay? And that test basically quantifies, I've always kind of related it to how pregnant are you. Like if you're working in hematology and they do a pregnancy test on a female that comes to the ER, and if you report that test out as positive, you better just get ready, because now they're gonna draw a blood sample and they're gonna do a beta HCG. And that beta HCG, you already know they're pregnant. Now, that test can kind of tell you how far along they are in pregnancy. It quantifies the pregnancy test. Anytime there's abnormal chromosome analysis or history of genetic disorders, that can also be an indication of why your gynecologist would want to do an amniocentesis. Of course, abnormal ultrasounds, if there's anything that looks wrong with the baby, it says fetal body measurements, that could also be an indication of why they would do amniocentesis. And in later pregnancy for possible early delivery, they'll do that amniocentesis to make sure that the fetal lung maturity is going on, and it's got those lung surfactants that's needed. Also, if they are detecting hemolytic disease of the newborn. Now, even though this doesn't happen very often, we're going to talk today about bilirubin levels, because if a baby truly does have hemolytic disease of the newborn, in other words, that baby in the womb, the mom has antibodies, they're crossing the placenta, they're causing the baby's red cell to rupture prematurely, causing the hemolysis to occur. We detect that through bilirubin levels. Remember, all bilirubin is a breakdown product of hemoglobin, right? So one of the tests that we can do for hemolytic disease of the newborn, we are looking for unconjugated bilirubin, all right? That's a test that we're looking for. It very rarely happens, but it's a big part of this chapter, okay? Tested for hemolytic disease of the newborn. If you actually collect the epithelial cells or the fetal epithelial cells, that's what they're doing all the genetic testing for when they're worried about a baby that has chromosome abnormalities. Now they use some words here, I won't test you, but this is some of the different testing they will do on those fetal cells. You may have ran across some of these words when you were doing your DNA papers or molecular diagnostic papers. Karyotyping, they can do the FISH test, fluorescence mapping, karyotyping, and DNA testing. Usually when they are testing for some of these substances, they will do a procedure called thin layer chromatography. That's in your chemistry book if you want to read about that. We talked about it briefly. All right, the collection of the fluid itself is called amniocentesis. It's where a needle goes into the amniotic sac and obtains fluid. It says the maximum amount you should take is about 30 milliliters in a sterile syringe. It says to discard the first 2 to 3 drops because they may be contaminated. These specimens you want to collect, you want to protect from light, because one of the major tests they're going to do is test for bilirubin, right? We all know light will degrade bilirubin. So now they're going to collect it in a tube bigger than this, but remember they make amber colored tubes that you can put this fluid in. They even say they make black plastic tube covers. You want to perform all handling procedures immediately and deliver to the lab promptly. Again, you guys probably would not be in on this about delivering it to the lab, but you need to at least have kind of a history about it. If they are doing fetal lung maturity tests, you want to put the specimen on ice, refrigerate it or freeze it up to 72 hours if needed. If they are doing cytogenetic testing, the specimens are to be kept at room temperature. Now again, we're talking about the amniotic fluid. It says centrifuge or filter fluid for chemical tests to remove debris. Generally, they are only going to filter the test if they are filter the fluid if they are doing fetal lung maturity tests. That's what that FLM stands for. Color and appearance. Normal amniotic fluid should be colorless with slight to moderate turbidity from some of the cells. I've already looked at your exam. There are questions about the colors of the amniotic fluid, so make sure that you have that marked down in your book. All right. If the fluid is blood streaked, this could just be from a traumatic tap where they went in to obtain the fluid, no problem, right? But it could be from abdominal trauma or, unfortunately, intra-amniotic hemorrhaging. Remember, there is a test that you can do to determine fetal blood versus mom's blood. You guys remember that Jason told you guys about a Clyhauer acid Becky stain?
Mhm.
If the fluid is dark green, this could be due to meconium, and that is the baby's first bowel movement. Okay, so if there's a little bit of stool in the fluid, it'll be dark green. You don't want to see a dark red brown color because that could be due to fecal death of the amniotic fluid. Again, make sure you know those colors. There's a chart right here basically that goes over what we just said. All right, hemolytic disease in the newborn. This should be very familiar to you guys because this is really brief and you guys have already covered this in blood bank. Babies that have hemolytic disease of the newborn are most commonly due to moms that are Rh negative. Okay, we're going to get into the Rh negative mom giving birth to an Rh positive baby. Okay, there are other red blood cell antigens that can produce hemolytic disease of the newborn, but remember when we say a mom is Rh negative, we're talking about the D, the anti-D, right? They do not have the D antigen on the red blood cells. So for anybody in here that's female that is Rh negative, I mean, you won't have any issues with it, but a long time ago you could have had issues. Basically, it takes a mom that is Rh negative that gives birth to 2 Rh positive babies back to back. Okay, that's how it works out. First baby, no problem. Second baby, there'll be problem if they don't administer the Rogaine. You guys remember covering that?
Yep.
The main problem that causes all of this is maternal antibodies will cross the placenta and destroy fetal cells with the corresponding antigen. If that mom develops an anti-D antibody that's floating around in her serum, and then she delivers another Rh positive baby, the entire time of that pregnancy, those antibodies are crossing the placenta and they can be coating the baby's red cells and causing the red cells to bust open. That makes sense to everybody? That produces hemoglobin, that breaks down to unconjugated bilirubin levels, and that's when we're testing for bilirubin. So here's a nice little picture. You got the mom, baby number 1. The mom, first of all, has to be Rh negative. What percentage of people are Rh negative? You guys need to know that.
Oh.
I mean, are we talking about something that happens in half the moms? Are we talking about something that just rarely happens? So what percentage of people are, in other words, what percent of people are negative? A negative, O negative, B negative, AB negative?
Less than 50.
What's that?
Less than 50.
A lot less than 50.
Like 20?
15%. 15% of the population is Rh negative. Okay? So most of you in here probably, if we're playing the odds, are Rh positive. You're not Rh negative. But for the ones that are Rh negative, okay, you have to think about this. Okay? Alright, so the mom's Rh negative. Baby number 1 is Rh positive. During delivery, there's a little bit of mixing of the blood. If that mom and that baby's blood mix up and there's more than the normal amount of bleeding, okay, that can cause the mom to be stimulated. Since she is Rh negative, that Rh positive blood mixes with her, and then she's going to develop this antibody. Here's the antibody that she develops. Look, the baby's already been born. Baby number 1 is fine, okay, no problem. But because of that delivery, she was stimulated. She developed those IgG antibodies. Remember, IgG antibodies are the only ones that can really cross the placenta, right? Remember, the IgM antibodies look like a big snowflake. They got 10 binding sites. IgG are small. They're small enough they can cross the placenta. So baby number 2, if baby number 2 is Rh positive, if baby number 2 is Rh negative, you got lucky, right? But if baby number 2 is Rh positive, these antibodies can cross the placenta during pregnancy and cause that baby to be born very anemic because the red cells are being lysed from the antibodies. That makes sense to everybody, kind of? Okay, so if the damage is done, unfortunately, and that's not the only thing that can cause the bilirubin to be elevated. There's other things, but that's the one that most textbooks want you to be concerned about. So bilirubin from red blood cell destruction will appear in the amniotic fluid. So what color is the fluid going to be if there's a lot of bilirubin in it?
Yellow.
Yellow. I think it says bright yellow. on that chart. It says the amount of unconjugated bilirubin present correlates with the amount of red blood cell destruction. Now I've got to talk about a test, and once again, I haven't done this test, but it does make sense on this. They will take a spectrophotometer and measure the absorbance or the optical density at a certain wavelength. You know when you guys use the spectrophotometers and you set it at a wavelength, right? It says spectrophotometric analysis of fluid optical density between 365 and 550 nanometers is plotted on semi-log paper. Bilirubin, if there is bilirubin in that fluid, the optical density rise will occur at 450 nanometers. So I want you to think they've got this fluid on a spectrophotometer and they're testing the absorbance between 365 and 550. About halfway through that, at 450, if there's bilirubin in that fluid, there's going to be a dramatic increase. In other words, 450 nanometers is the perfect wavelength to detect bilirubin. Okay? So it says bilirubin causes the optical density to rise at 450 nanometers. The difference between baseline and 450 peak is what's called the change in absorbance. The difference can be plotted on a Liley graph. Okay, so here's the deal. If you're testing normal amniotic fluid that doesn't have bilirubin in it, okay, and you're using a wavelength between 365 and 550, you're going to get a line that basically starts at about 0.5 absorbance value, but see how it's going to go down. It's going to make a straight line and go straight down. If there is bilirubin in that fluid, once it's at 450 nanometers right here, there's going to be a sharp increase in the absorbance of the fluid, okay? That's where they can detect if bilirubin is there. So basically, you don't want a change in absorbance at 450. You want it to be on this normal line right here. Let's say this amniotic fluid right here has no bilirubin in it, okay? So when you go through these different wavelengths, it's basically going to make a straight line gradually going down. But if there's bilirubin in that fluid, once you are testing it at 450 nanometers, there's going to be a jump, okay, in the absorbance. You determine how much bilirubin is in that fluid by the difference in the jump. If there's no jump, then it's normal fluid with no bilirubin. In other words, it's going to be plotted probably on this normal curve right here. But if you've got a big difference, let's say the fluid, instead of being at 0.3 where it should be, it jumps up to 0.5, that would be a difference of 0.2. That would be a change in absorbance at 450 of 0.2, all right? And that can indicate problems. As you're going to see, we're going to present this on a graph. This is called a Liley graph, is what we're talking about. This Liley graph, basically you plot the change in absorbance at 450 nanometers to the gestational age of the baby. It consists of 3 zones based on the different varying amounts of hemolytic severity. If it falls in zone 1, it means usually the fetus is only mildly affected, all right? That basically means there's not a lot of bilirubin there, but there's some, okay? If it falls in zone 2, that requires careful monitoring of the baby. And if it falls in zone 3, that means the fetus is severely affected, okay? That means there's been lots and lots of red cells lysed. It may require induction of labor or an intrauterine exchange transfusion. This is not good when this happens. I never had to do one, but there is actually a way that they can transfuse a baby in the womb with blood products, okay? Now, I'm assuming that would more than likely always be done at a place like Children's, but in an emergency situation, it might be done at CHI. Okay, we had a procedure manual that went through it, but luckily I was never in blood bank when we had to go over it. So here's that Liley graph that I was talking to you about. You remember that example that I told you about? For example, at 450 nanometers, if it's supposed to, if the absorbance reading should be at 0.300, but you get 0.500, you agree that would be a difference of 0.2, right? So if we looked on this graph, look where 0.2 falls. It doesn't matter what the gestational age is. If it's a difference of 0.2 at any gestational age, that falls in zone 3. Zone 3 is not good, right? That means that basically, if it falls in zone 3, you gotta start looking at the lungs of the baby. Are the lungs mature enough to induce labor early? That's where all of this kinda, you should be able to kinda correlate it. You test for bilirubin to see if there's hemolytic disease of the newborn. If it's bad, now you've gotta start testing for fetal lung maturity to see if that baby can be delivered. That's most of the time why babies really struggle, is their lungs don't work good, okay? That's why they gotta be real careful about early deliveries. Is that baby's lungs mature enough that it can survive outside? Okay, so there are tests that are done for fetal distress. Now, I'm not talking about hemolytic disease of the newborn here, okay? I'm talking about defects that are going on with the baby called neural tube defects. These are gonna involve things, I'm sure you've heard of these words before, but you've probably heard of anencephaly or spina bifida, okay?
Yeah.
Those are things that are very bad, okay? And a lot of times, these are called, they have a word for it, it's called neural tube defects. It's where basically you got a liver, and if the spinal column or around the brain area, if it doesn't close, this is very early, like within the first week of gestation. If that doesn't close, all right, there is gonna be a substance called alpha-fetoprotein that basically seeps out of the baby because there wasn't a closure. Think about right here, okay, in a really small baby, it's open, okay? If that doesn't close up, there's things that's inside the baby that kind of leak out into the fluid, and that indicates neural tube disorders. I would not recommend you go under Yahoo Images to look at that, because it's really bad, okay? If those babies do come to term, it's not good. Okay, so neural tube defects. Alpha-fetoprotein is produced by the fetal liver prior to 18 weeks gestation. Increased levels of maternal blood or amniotic fluid indicate possible anencephaly or spina bifida. Just correlate those two words with neural tube disorders. It says increased levels are found when the skin fails to close over neural tissue. But don't make the mistake, if you see this on a registry exam, this AFP or alpha-fetoprotein is usually tested on the mom during a prenatal visit, okay, to the gynecologist or the OBGYN. Normal values are based on the weeks of gestation for fetal distress. They can be reported in multiples of the median. The median is the lab's reference values for a given week of gestation. More than 2 times the median is abnormal. You can follow this test with something called amniotic acetylcholinesterase. This test is actually more specific for neural disorders, but you don't want to do this test on a bloody specimen because blood does contain acetylcholinesterase, so you might get a false positive. So any test you see that has to do with AFP or multiples of the median or acetylcholinesterase, those are tests that have to do, unfortunately, that the baby could have neural tube disorders, all right? Because we're fixing to have to start talking about different tests. The thing about this chapter, I think, that makes it hard on students is you have to basically, when you're studying your notes, you have to determine, okay, these tests are done for bilirubin, okay? These tests are done for neural tube disorders. These tests are done for fetal lung maturity. So you gotta be able to separate those 3 different conditions. Because when you first read this chapter, it's kind of hard to keep it all straight. All right, fetal lung maturity. This is the most common complication of early delivery. Another name is respiratory distress syndrome. It usually occurs because of lack of what's called lung surfactants. Lung surfactants are responsible for keeping the alveoli open during inhaling and exhaling. Surfactant, what it does is it decreases the surface tension on the alveoli so they can inflate more easily. Many lab tests are available for fetal lung maturity. The first one, or the gold standard, is called the lecithin-sphingomyelin ratio, or the LS ratio. You will probably never hear it called actually lecithin-sphingomyelin, but that's what the L and the S stand for. Everybody with me? It says lecithin is the primary component of lung surfactants. Increased production occurs after the 35th week of gestation. Sphingomyelin is produced at a constant rate after the 26th week of and serves as a control for the rise in lecithin. So an LS ratio is usually 1.6 prior to week number 35, but after week 35, it will rise to 2, okay? So you want it to be at 2. Does that make sense to everybody? Because if it's at 2, it means that more than likely, there is going to be enough lung surfactant that that baby can survive. It can breathe on its own. So it says, therefore, preterm delivery is considered safe with an LS ratio of 2 or higher. Remember that. This test is usually used using thin layer chromatography. It does say many labs have replaced the LS ratio with the quantitative phosphatidylglycerol immunoassay and lamellar body density procedures, and we're fixing to talk about those tests as well. But I still, every book I've ever seen, the first test they talk about, when you're talking about fetal lung maturity, they talk about the LS ratio. Okay, phosphatidylglycerol. This is a lung surface lipid that is also needed for lung maturity. It will usually parallel lecithin, except in diabetic moms. But remember, there is something that occurs in a lot of moms called gestational diabetes. Remember us talking about that? A lot of expectant moms, I'm not worried about you knowing the exact why, but when they get pregnant, they basically turn into diabetics for the length of the pregnancy, okay? And when they deliver the baby, the diabetes goes away, okay? That's called gestational diabetes. So you have to also do this test for phosphatidylglycerol. You pretty much have to do it on all moms because you don't know if they are in gestational diabetes. But if they do have gestational diabetes, this particular lung surfactant kind of lags behind. If they don't have gestational diabetes, it will parallel the LS ratio. There is a test called an amniostat fetal lung maturity that is an immunologic agglutination test for phosphatidylglycerol using antibodies specific for the glycerol that can replace the LS ratio. Blood and meconium do not interfere with this test. Lecithin and sphingomyelin are also in blood, so you want to be careful when you're doing that test on a bloody specimen, okay? That would cause, again, a false positive. There is a foam stability test. This can be performed at the bedside. When you read about this in your textbook, it's real easy. Like when they're fixing to deliver, okay, or they're looking at this fluid, they can take the fluid and they can basically put it in a tube and shake it. Okay, it's called the shake test or the foam stability. If they shake it and bubbles are produced, that's a good thing. If no bubbles are produced, that's not a good thing as far as the lung maturity. That's a real quick test. Now, I'm fixing to have to talk to you about, they've done it now to where they don't just shake it and look for bubbles. They actually do kind of like a serial dilution on the fluid about just how much alcohol can make it either have bubbles or not have bubbles. That's called an index. So it says amniotic fluid can be mixed with ethanol, shaken for 15 seconds, and allowed to sit undisturbed for 15 minutes. A continuous line of bubbles around the outside edge indicates that there is presence of enough sufficient amount of surfactant to maintain the alveolar stability of the lungs. Alcohol is an anti-foaming agent, and fluid can overcome this if there's enough surfactant in it. That's going to be a quick test thing, all right? But then they start talking about the foam stability index, where basically they mix the fluid with increasing amounts of alcohol. See what I mean? And that way they can kind of quantify just how much lung surfactant is present in the fluid. So you can read about that. But I kind of think of that as kind of like a serial dilution using alcohol. lamellar bodies. Lamellar bodies are the storage form of surfactants. They contain approximately 90% of phospholipids and 10% protein. It tells you that pneumocytes produce these, what's called lamellar bodies. They will increase in amniotic concentration from 50,000 to 200,000 per mil by the end of the third trimester. Tells you what the optical density of them are. I'm not really interested in that. I am interested in you realizing that the lamellar body's diameters, they range from about 1.7 to 7.3 femtoliters. I'm not going to test you on any of those numbers, but basically what I want you to know is you can take amniotic fluid and you can run it through a CBC analyzer and it will count those lamellar bodies as platelets, okay? So if you wanted to do a real quick lamellar body count, you could basically go over to whatever analyzer runs CBCs, all right, and run it through that machine, and don't pay attention to anything but the platelet count, because the platelet count will tell you how many lamellar bodies are in that fluid. Isn't that pretty neat? I mean, it's just a quick way. And if lamellar bodies indicates the storage form of surfactants, that can correlate with fetal lung maturity, all right? So you can read about all of that, but basically that's what I want you to know about the lamellar body count. It goes on to tell you about running it through the analyzer and all of that, putting it on the rocker. I just want you to know that you can get a lamellar body count by actually running it through the platelet channel on a CBC analyzer. Any questions on that?
Any of those questions that you can?
Yeah, let's go through some of this first. I almost forgot. I hope it didn't interesting over. Oh, bear with me, I gotta look at it.
Excuse me.
Okay, number 2, what is the primary cause of the normal increase in amniotic fluid as a pregnancy progresses?
Fetal. C.
What's that?
C.
Number 2. Number 2, what's the primary cause of the normal increase in amniotic fluid as a pregnancy progresses? Fetal urine, C. Is that what you said?
Yeah. You gotta be louder.
Number 3, which of the following is not a reason for decreased amounts of amniotic fluid?
A.
A, fetal failure to begin swallowing. Remember, we said one of the reasons that you can have a decreased amount of fluid is the baby's swallowing too much amniotic fluid, right? Or fetal urine. Number 5, amniotic fluid specimens are placed in amber-colored tubes prior to sending them to the lab to prevent the destruction of which of the following substances? Bilirubin. Number 8, definitely need to know that matching question. Match the following colors of amniotic fluid with their significance. What do you think colorless fluid indicates?
Normal.
Dark green.
The presence of the first bowel
Remember meconium, the
Yes.
baby's first bowel movement? That's
Yep.
what meconium is. What about red-brown?
That's
Death.
death.
Good, fetal death. And then yellow.
Bilirubin present.
Good. Okay, number 9, a significant rise in the optical density of the amniotic fluid at 450 nanometers indicates the presence of which analyte?
Bilirubin.
A, bilirubin. That's where I was telling you. I mean, I know it's going to take you guys studying a little bit, but you need to associate, like, bilirubin has to do with hemolytic disease of the newborn, okay? And so that optical density test, remember that Liley graph we looked at 450 nanometers? So the answer to number 9 is A, bilirubin. I don't expect you to get this yet, but I do expect you to know it before we take the test. Number 10, see, plotting the amniotic fluid optical density on a Liley graph represents severity of hemolytic disease in the newborn. A value that is plotted in zone 2 indicates what condition of the fetus? C, moderate. Good, C, moderately affected fetus that requires monitoring. Number 11, the presence of a neural tube disorder may be detected by which of the following tests?
Alpha fetoprotein.
Good. B, alpha-fetoprotein. Remember, that detects neural tube disorders. And again, when we're studying this chapter, I would break it down, just go through the chapter and go, okay, what test did he say had to do with bilirubin? Okay, what test did he say had to do with fetal lung maturity? All right, and what test had to do with neural tube disorders? Like, break it down into 3 sections, and that'll cover all of them. Number 18, this is good. Does the failure to produce bubbles in the foam stability index indicate increased or decreased lecithin?
Decreased.
Decreased. Hold on, I gotta think about this one. Yeah, decreased, the failure to produce bubbles. Number 19, the presence of phosphatidylglycerol in amniotic fluid fetal lung maturity test must be confirmed when, remember that one test I told you about, the PG phosphatidylglycerol? B, the mother has maternal diabetes. Remember I told you that one kind of stays back if the mom is in gestational diabetes. Number 14, true or false? Prior to 35 weeks gestation, the normal LS ratio is less than 1.6. That's true. And it jumps to 2, right? And that's when it's safe to deliver the baby usually. Number 4, back on page 251, why might a creatinine level be requested on an amniotic fluid? This is number 4.
Determine if it's like the maternal urine.
Yep. C, to differentiate amniotic fluid from maternal urine. Just always remember, guys, urine will always have the highest amount of creatinine. Okay? There's not a fluid in your body that will contain creatinine more than urine does. Okay? So that's one way they can distinguish. If they don't know if it's urine or spinal fluid, even if they ask you, how can you tell urine from spinal fluid? Okay, let's just say somebody had a real clear urine that looked like spinal fluid and they couldn't tell the difference. There will be creatinine in that urine specimen, whereas there won't be in that spinal fluid. Okay? Most fluids will never contain the amount of creatinine that urine does. All right. It's 9 o'clock.
Yep.